Hey everybody. So I am recording this sort of mini lesson for those who aren't around this week in class. If you haven't checked your email lately, I am back Tuesdays and Thursdays, but if our signals get crossed and you happen not to be able to get in on Tuesday and Thursday for whatever reason, like the 8 of you that missed my class for an AP test yesterday, I'm sure you rocked it. Then this will be kind of a good substitute, and it'll give you the same background into blood typing that I gave to everybody else. So, without further ado, let's jump right in. So, I figured it was kind of interesting and relevant to dive a little bit into the genetics of blood typing. And most traits that you would pass on to your offspring are controlled by two alleles, which are just different forms of a gene. When Gregor Mendel did his famous experiments with pea plants, there were alleles that gave you short plants and alleles that gave you tall plants. In this case, it's slightly more complicated, but not really, because blood type is controlled by three different alleles. In this case, we have IA and IB, and you'll notice that those are capital I's to denote those are dominant traits. If you get the IA allele, you will get the A antigen on your red blood cells. If you get the IB allele, you will get the B antigen on your red blood cells. And these two are co-dominant, which means that if they show up together, if you get both different dominant alleles, you will get both dominant traits. Now, little I here shows a recessive trait. Now, a recessive trait is one that you would only exhibit if you had both copies of that allele. So your mother and your father would both have to give you that recessive allele in order to get that particular trait. Now, when I talk about antigens, I think that bears a little explaining. So let me show you a little picture here, and this will kind of clear up what we mean here. Now, an antigen is what we call, in this particular case, the blood group antigens are what we call glycoproteins, mixtures of proteins and carbohydrates that are used as markers for your immune system. So if my mother, for example, had type A blood, and that means that all of her red blood cells contain the A antigen on their surface, and her immune system recognized only blood with the A antigen as belonging to her, and she would have antibodies against anything that wasn't the A antigen. Now, the B antigen would show up in someone with type B blood, and that is a different glycoprotein that the immune system of someone with type B blood would recognize as their own. If you have type AB blood, that means you have both of those antigens, both A and B, on the surface of your red blood cells, which means you would get both dominant alleles. And if you have type O blood, like I do, then that means that you would have no antigens on the surface of your red blood cells because you would receive both recessive alleles. But back to the genetics. So if you have type A blood, like we said before, you have the A antigen on your red blood cells, and you have the anti-B antibodies, that is, antibodies against the B antigen, pre-formed in your plasma. That means from birth, from day one, you've got antibodies against the B antigen. And that will allow you to fight off that B antigen as though it were a foreign invader if it enters your body. In the context of the immune system, that's a very good thing. Those of you who have gotten the COVID vaccine, I got my second dose about 3 weeks ago, you would have built up antibodies upon exposure to that infectious agent, that antigen, if you will. And when you're exposed to COVID again, you would fight that off as though it was an invader, because it is. In this case, these antibodies don't have to be formed in response to exposure to those antigens. They're already there. So if 3 days after birth you had to get a blood transfusion, hopefully this would never happen, but if you were to get a blood transfusion as a very, very young person, you would have antibodies against that antigen. So, the possible combinations of alleles, of different forms of that gene that would get you that blood type, would be either two dominant alleles, both of which carry the A antigen, that is, your homozygous dominant, or your heterozygous, which means you have two different alleles, one dominant and one recessive. It turns out that that would be the combination that my mother has for reasons which I'll talk about in a little bit. So then we have type B blood. In type B blood, we have B antigens on our red blood cells, and we have antibodies against the A antigen that are preformed in the plasma, that is, present from birth. And to get that blood type, you could either be homozygous dominant, you could have both B alleles, or you could have the dominant allele for the B antigen and a recessive. If you have type AB, you have both antigens on your red blood cells, and you have no antibodies in your plasma. Now, from your standpoint, that's a very good thing because you are what's called a universal recipient. If someone donates blood to you, anybody can give you blood because you don't have any antibodies that will trigger that immune response that will cause that blood to clump up if foreign antigens enter, because you have all the antigens. Of course, since these two alleles are co-dominant, the only way you're going to get type AB blood is if you get 1A and 1B. Now, type O is in a tough spot from a recipient standpoint, but people like me that are type O, and like my father as well, we can donate blood to anybody. We don't have any antigens on our red blood cells, so the only blood that our body recognizes is other type O blood. So we can only receive type O blood because we have anti-A and anti-B antibodies in our plasma. And so we will not cause anybody else to react to our blood because we don't have any of those surface antigens, but we will react to any of those antigens that come into our body. And of course, that is the recessive trait. So in my case, my mother was A positive, my father was O positive. Both of them gave me that recessive allele to make me O negative. Now, we'll get into what that negative means in just a minute. Actually, let's talk about that now. So the Rh antigen is the thing that makes you positive or negative. And that is sort of similar to the way that your immune system works in being exposed to a virus or a bacterium. When you got the COVID vaccine, you started building up antibodies against the COVID-19 virus. Or if you were previously exposed to the COVID-19 virus, if you got sick at some point in the last year, then your body built up antibodies. So if you got reinfected with COVID, you would either have a very mild case or those antibodies would be able to fight it off entirely. That's the whole idea behind vaccines. You're building up your body's immune response. In the case of the Rh antigen, you don't have antibodies preformed in your plasma against the Rh antigen if you don't have it. If you're exposed to Rh positive blood, so if my dad, who was O positive, gave me blood, then I would build up antibodies against it. And so I might get a little sick. I might feel run down for a couple days, but then I'd be fine. If I was exposed again, the same kind of clumping reaction would occur that occurs when you're exposed to the other antigens, A or B. Now, I think in the interest of time, I'm actually going to cut this short to make these nice bite-sized videos, so hopefully that gives you some background into the genetics of blood typing as well as what antigens and antibodies mean in the context of blood donation. In the next video, I'm actually going to go through a simulated blood typing experiment to show you what it would look like. So, I hope you enjoyed this, I hope it was useful, and have a great day.